Hello and welcome. You are listening to a podcast by the Milner Centre for Evolution at the University of Bath. I'm Professor Turi King, your host, and today I'm talking to Corey Holt, whose research focuses on microbial diversity and host-microbe interactions in the marine environment. Corey, I'm going to start with the very basics. For those listening who don't know, what are microbes and why are you interested in how they interact with their host?
Yeah, so microbes basically encompass most of the life that we know it. They're tiny microorganisms that we see under the microscope and come in various shapes and sizes and do various functions. I'm generally interested in what's known as the microbiome, so the community of microbes that kind of live with a particular host or with an environment. And so I'm interested in the identity of those microbes, but also their interaction. So how they're interacting with each other and how they're interacting with the host. In some cases, those interactions can be really important, indeed vital for the host's success. So whether it's kind of supporting the immune system or providing access to a previously inaccessible source of nutrition.
So which hosts are you looking at?
So I tend to focus on marine invertebrates. So if I were to ask you to think of an animal, you might think of a cat or a dog or something fluffy, but most animals are very small marine or aquatic invertebrates. But when we look at kind of microbiome research and kind of characterizing those interactions, it's dominated by mammals. And so my aim is to explore some of those perhaps less popular animals that still have really important roles in the environment, whether it's kind of important positions in the food chain or whether it's kind of contributors to overall ecosystem health, and try to explore those host microbe interactions in those really important but often overlooked animals.
So tell me about these animals. What ones are you looking at?
Yeah, so it's quite a mix. I actually started as a PhD student looking at lobsters and did a bunch of work on kind of lobster microbiomes and parasites. But now it's more general. And so I'm not particularly focused on one animal, but I'm interested in kind of what determines these microbiome composition and interactions across an invertebrate diversity and looking for kind of broad trends. So some of those might be really small invertebrate crustacea called copepods. Some of them might be anemones. Yeah, we've done all sorts.
So how do you look at this?
Yeah, so we use a number of different techniques. It kind of depends on what microbe it is that we're interested in. We often think of the microbiome as purely bacterial, but actually there's lots of different microorganisms within the microbiome. So archaea, viruses, or fungi, or kind of the other one that I focus on is protists. So these kind of unicellular eukaryotes. So we typically use something called metabarcoding to characterize those communities. And essentially, if you think about kind of shopping in a supermarket, every item has a barcode that identifies that item. And it's kind of the same when we do that microbiome sequencing in that we can look for DNA barcodes that identify microbes within kind of a homogenate of the microbiome and then separate it out after we sequence them. And so we do that generally for the microbiome as a whole. When we focus on particular organisms like protists, we try and add some microscopy, and so we can actually get an idea of what they look like and what they're doing.
So talk to me about these protists. What are protists?
Yeah, so protists are any eukaryote that isn't a plant, animal, or fungi. That might seem like a bit of an unsatisfying definition, but it's actually quite a helpful one. There's some discussion about whether that should be unicellular eukaryote, but there are some unicellular eukaryotes that have multicellular stages. And so let's stick to any eukaryote. We have that definition because, like I said, they're incredibly diverse, both in form and function, but also in terms of evolutionary history. So protists occupy the majority of the eukaryotic tree of life, and they aren't what we refer to as a monophyletic group. So they aren't all the descendants of a shared ancestor and nothing else. And that's because that group includes animals, plants, and fungi. So historically, we've thought of protists as kind of a kingdom. So back in 1818, we get this description of the protozoa, which was used to include some of the protists we know today, but also so-called simple animals. So that's where the name comes from, kind of first animals. And then later, we get the protista, this kingdom that was used to separate protists from animals and plants. That prevailed for much of our history, including in Whitaker's very famous five-kingdom hierarchy. But now, with the advancement of kind of DNA sequencing and with characterizing ultrastructure, we realize that protist diversity is the bulk of eukaryotic evolution, and animal, plants, and fungi are just branches within that diversity.
So when you're doing your sort of barcoding, are you essentially trying to look at, okay, so we've got these particular organisms seem to be in this particular environment, these ones seem to be in this particular environment, and you're trying to understand like why that's the case?
Yeah, absolutely. So that's kind of one of the things that I'm really trying to focus on is what determines those compositional differences, whether that is the environment, as you mentioned, or whether that is something to do with the host biology. And in our animals, we actually find that it's the environment that has a big say in what grows within these organisms. Whereas if you were to look at mammals, some other terrestrial animals, it tends to be that more similar hosts have more similar microbiomes. It could be a number of reasons for that. Our animals live in the water, right? So they're swimming through this microbial soup, and perhaps there's kind of less say in what establishes within those communities. But it's really important for us to understand kind of the dynamics going on here, because the composition of the communities will determine the kind of interactions, and that can have a big impact on host health and fitness.
And what's the wider importance of this? Is it, food security, understanding the food chain, that kind of thing?
Yeah, that's one of them for sure. So lots of people might use this sort of work to identify ways in which they can improve health through kind of supplementation or manipulation of that community. I'm personally interested in food security. And so over half of the seafood we eat comes from aquaculture, so farming aquatic protein, whether that's on land or in kind of sea-based facilities. But disease is a kind of major barrier to developing that process in a more sustainable way. And so if we can understand kind of the interactions that are going on in the microbiome, we can perhaps prevent some of that disease, but also kind of improve the general health of those animals so that they can deal with the disease.
So what you're looking at is basically how organisms are interacting with one another. And I know one of the things that you're quite interested in is parasitism. So tell me about that and what you hope to find out.
Yeah, so there are lots of parasitic lineages and even groups within the protist diversity. And some of those we've identified in our microbiome screening. And often they kind of dominate those microbial surveys. But remarkably, due to the nature of that sequencing, where it's a very short sequence and you can't really get anything more from that sequence in terms of its function or what it might look like, that means we don't really know what a lot of them are actually doing or indeed look like. And so what I hope to do is use those surveys to guide the isolation of some of those parasites. And so there's a couple of groups that I'm interested in. So one of them is the apicomplexer, which you might not have heard of, but you'd almost certainly know of some examples of apicomplexan. So Plasmodium is the causative agent of malaria. That's an apicomplexan. Not all of them are parasites. Some of them kind of just live in the gut, but many are parasites, and we find them in the sorts of animals that we look at. And then the other is a group of dinoflagellates called the marine alveolates. So dinoflagellates are quite famous in that one of them, or actually it's now a group of them because it's got reclassified, but a group of them live with coral and photosynthesize. So we often call them zooxanthellae or photosynthetic algae. These are dinoflagellates. And there's a group of really abundant, really prevalent dinoflagellates, these marine alveolates. But again, we know very little about them. They just pop up in our microbiome surveys a lot. So yeah, we're hoping to isolate those. The ones we do know infect quite important hosts, whether that's kind of fish in aquaculture facilities, whether that's some algae that form harmful blooms in the ocean. And by using these techniques of microbiome sequencing, of microscopy, and better understanding those interactions, we are able to perhaps identify kind of mechanisms of parasitism, and maybe we're able to target those mechanisms, or maybe we're able to identify those mechanisms in other organisms that have the potential to impact food security. So it's kind of coming at it in both angles, both as a preventative measure, but also kind of treatment as well.
I know your research generally falls under the idea of One Health, so that human, animal, environmental health, they're all interconnected, and therefore changes to one can have far-reaching consequences. So talk me through that.
Yeah, that's kind of the general idea of One Health. So typically we had this idea that there's kind of a one pathogen, one disease model, but actually we now have a greater appreciation that disease and kind of health conditions are multifactorial, and it is not just the parasite that is causing the disease, but it's perhaps the environment in which you find yourself in. So my work looking at kind of the diseases of these invertebrates tries to consider kind of what is the microbial community in the environment, how is that impacting that animal. And often they kind of relate back to human health as well. Although I don't focus on human health, you can imagine how a disease in something we rely on for food might have quite vast implications on us as humans, as consumers, but also as producers. And so there are lots of different examples where you can draw these kind of through lines between the three domains. And I think more comprehensive treatment methods that encompass those three domains will ultimately prove much more successful.
So talk me through one of those through lines.
Yeah, so let's think about coral. So as I mentioned, coral harbor those photosynthetic dinoflagellates. They belong to a group called the Symbiodiniaceae. And coral bleaching is when these coral actually expel those dinoflagellates and they're no longer able to photosynthesize and they'll die. And that's kind of a process which is driven by human-induced climate change. Coral are really important for environmental health. They have key functions in kind of ecosystem functioning and ecosystem services. They're key nursery grounds for other animals within the environment. They also are drivers of tourism for humans. And so if we as humans are negatively impacting the health of corals, it not only has a direct impact on the health of those animals, but also the health of the environment around it, and ultimately us as humans who rely on kind of the provisions that that ecosystem provides and kind of the tourism that it supports in many places.
So what excites you about what you're working on? What is it that gets you up in the morning?
So for me, it's the fact that we have this mass of eukaryotic radiation in the protists, yet many of these groups and lineages remain kind of uncharacterized or lack sequencing data. And when you get into that sequencing data, you start to uncover really interesting stories in terms of kind of the adoption of parasitism, which is something we've looked into, or kind of the loss of photosynthesis. And our interpretations of these kind of major events in history is changing every year. And to be able to kind of work in that field and be able to ask those questions, I think is really exciting. And then on the other side, I really just want to find new protists and find new protist parasites. And so lots of my time is usually spent under the microscope, where we're kind of sifting through seawater and seeing weird looking cells and thinking, oh, what's that? And then we isolate it and sequence it. And if I can do that for the rest of my life, then I'll be happy.
Corey, thank you so much for talking with me. This was a podcast by the Milner Centre for Evolution at the University of Bath. I'm Turi King, and thank you for listening. If you have any thoughts or comments on this or any other episodes, please contact us through social media. For more information about the Milner Centre for Evolution, you can visit our website.